Okay, welcome to the video lecture for Chapter 4 on carbon and the molecular diversity of life. Primarily in this chapter, we're going to focus on carbon-based compounds, also referred to as organic compounds. There are four categories of these: proteins, sugars, fats, and nucleic acids. We're going to focus in detail on each of those four categories in Chapter 5. But Chapter 4 sort of is meant to give you a primer of the basic structure of these molecules, okay? And the basic structure has to do with the atom carbon. If you take a living organism after death, rather, and you dry out the body entirely, what you end up with is about 50% carbon, or 50% of the weight of that mummified body basically is composed of carbon. Life on Earth is referred to as carbon-based, and this is because carbon forms the structural framework or structural backbone of proteins, sugars, fats, and nucleic acids. And the reason why it does this primarily has to do with its valence. Again, as we talked about in a previous chapter, valence refers to the number of bonds that an atom forms. In other words, how many electrons it's missing in its outermost shell, and therefore how many bonds it would need to form with other atoms to complete the outer shell. Carbon's valence is 4. It always forms 4 covalent bonds, and as a result of that, it's very good at serving as a framework sort of molecule, a framework atom, a structural atom for building molecules. And it bonds preferentially with hydrogen, oxygen, and nitrogen, as we talked about earlier, and as we'll see a lot more of in the upcoming chapters. So living organisms then consist mostly of carbon-based compounds, and because of its valence, carbon is really unparalleled in its ability to form large, complex, and varied molecules, okay? And so proteins, sugars, nucleic acids, fats, these distinguish living matter from non-living matter, but they're all composed of carbon compounds. All right, so take a look at this figure. Here you see a species of monkey that lives in the mountains of Japan. And obviously the monkeys are living organisms. They are all composed of carbon-based compounds or organic compounds. They are organic beings. But this is also true of a lot of the other things that you see here. Look at the moss on the rocks, or the bushes, the trees. All of that represents organic material, carbon-based compounds, okay? It's also true of things you can't see there, like the bacteria, the viruses, the single-celled protists living in the water, for example. These are all examples of living organic beings composed of carbon-based compounds, all right? So carbon-based compounds are ubiquitous throughout the living world. So if we take a look at a carbon compound here, we can see how carbon forms these bonds. So what you see in the center is a carbon atom covalently bonded to 1, 2, 3, 4 other atoms. And that structure is called a tetrahedron. We saw that earlier. Basically think of it as a pyramid, whereas a pyramid has a square base, the tetrahedron has a triangular base. If you draw lines between all of those atoms, you see that you end up with a tetrahedron, all right? And so carbon bonded to 4 other atoms forms this basic structure, this tetrahedral structure. That's a basic structural building block of organic compounds. You see it in DNA molecules, in proteins, in fats. You see it in sugars, all right? Okay, so what we're talking about here with regard to carbon-based compounds is organic chemistry. Now, organic chemistry is very, very complex. It's a very challenging subject. We're going to talk about it, though, in very, very basic terms here. Just enough organic chemistry for you to understand structures of molecules, essentially, okay? Now, organic chemistry is the study of compounds that contain carbon. And in particular, for a compound to be considered organic, it must, at minimum, contain carbon and hydrogen. As long as it contains carbon and hydrogen atoms, it's considered to be organic, all right? But it has to have at least those two. It can have other things, but it has to have at least those two. Now, organic compounds vary in size considerably. They can be very simple molecules. Very good example of one of the simplest ones is methane. Methane is CH4, has 5 atoms in it, 1 carbon bonded to 4 hydrogens. That's one of the smallest organic compounds, 5 atoms total. But compare that to a human DNA molecule, which could have upwards of several hundred billion atoms in it, and now you start to understand how large these molecules can be. And there are sugar molecules that are even larger than that. Some of the molecules that build cell walls of plants, for example, like cellulose. And we'll see those in the next chapter. So there's a lot of range and size here of these molecules, okay? Now, we can also have oxygen, nitrogen, phosphorus, sulfur atoms as well. Those are some of the more common atoms that you see in organic compounds. All right, so in terms of understanding the structure of organic compounds, we have to go back to electron configuration, right? Electron configuration, how many electrons an atom has, and in particular, how many it has in its outer shell, and where those electrons are in terms of the orbitals around the atom is the key to understanding an atom's characteristics, as we talked about in Chapter 2. Electron configuration determines the kinds and number of bonds an atom will form with other atoms. So, carbon has a valence of 4. That means that it has 4 outer shell or valence electrons. That's what it means indirectly. Directly, valence is referring to how many electrons are missing in the outer shell. So carbon having 4 electrons and the outer shell able to hold 8. Carbon is missing 4. Its valence is 4. It therefore has a bonding capacity of 4. It needs to pull in 4 more electrons, 1 in each of 4 covalent bonds with other atoms, to complete its outer shell. All right. And as we said, that in molecules with multiple carbons, each carbon bonded to 4 other atoms has that tetrahedral shape, all right? And so, for example, if you're looking at methane would have the structure carbon in the center, 1 hydrogen up here. It would have another hydrogen there, another hydrogen there, and then 1 sort of going back into the plane of the screen here, like that. That's how these are typically drawn, okay? Another way you could do that to show it as simply 4 single bonded hydrogens to that carbon, okay? Now, what if you had 2 carbons joined by a double bond? Well, in that case, we would draw it like that. Now, each of these carbons having 2 bonds to another carbon only has 2 more bonds it can form. And let's just make it simple. Let's say that they're the hydrogens. You would have a molecule structured like that. So, this carbon is double bonded to that carbon, 2 bonds there. Okay, it only has 2 more bonds it can form. In this case, those are the hydrogen. The same is true over here. All right. So, but you see that each of those carbons has 4 bonds. But also, if you think about it, hydrogen has a valence of 1. That first shell can hold 2 electrons. Hydrogen only has 1. It needs to form a single bond to complete its outer shell, and that's why each of those hydrogens forms 1 bond with the carbon. Okay, let's look at some other examples. So here we have methane, for example. I showed you that, CH4. There's the structural formula. This is called a ball and stick model. It kind of shows you the 3-dimensional shape of it, but you can see sort of highlighted there in that pink line is that tetrahedral shape. That's a space filling model, giving you a better idea of what it might look like. Now, this is methane. Here we have ethane, C2H6. So C2H6. Notice that each carbon is bonded to 4 other atoms. This one, 1 other carbon and 3 hydrogens. That's a tetrahedron. This one, 1 other carbon, 3 hydrogens. That's a tetrahedron. Here you see it. Here's 1 tetrahedron, and there's a second tetrahedron. It's as if the apices of those 2 tetrahedra are sort of bonded to each other by the bond between the 2 carbons. And this is the space-filling model showing you what that would look like. All right, here we have ethene. Note that separate ending, that different ending, ENE. I'll get to that in a minute. C2H4. This is what I drew in the previous slide. So carbon or two carbons rather, double bonded to each other, each one therefore having two hydrogens attached. That carbon has 1, 2, 3, 4 bonds to it. This one has 1, 2, 3, 4 bonds to it. Each of the hydrogens also has a single bond to itself. So because of that double bond, this molecule is actually two-dimensional. It's flat, as you see here. The double bond prevents those carbons from rotating relative to each other, so they end up in the same plane, as do their hydrogens. And here you can see the space-filling model of that. So organic chemists have a system of nomenclature, or a naming system that they use to name organic molecules. It's kind of complicated, but you see parts of it here, okay? Look at the endings: ethane, ANE, methane, ANE. The ANE ending means that all bonds to the carbons are single. All single bonds here, all single bonds there. The ENE ending that you see in ethene, or ethylene as it's also called, means that there's at least one double bond somewhere, and there you see it. There's also another ending, which I'll just write here, YNE, and that means you have a triple bond somewhere between two carbons. Okay, so the ending tells you something about the structure of these molecules. And there are other aspects of this naming system as well, but essentially the naming system works so well that an organic chemist can look at a long drawn out name of a molecule with words and letters and numbers in it. And if they understand the naming system, they can picture the structure of that molecule in their head. That's the value of it. All right, and we're going to see some aspects of that as we go along here. Okay, so the electron configuration of carbon gives it covalent bonding compatibility with many different elements. But in particular, the valences of carbon and its most frequent partners, hydrogen, oxygen, and nitrogen, are the building code for the architecture of living molecules. Let's see why this is. So if we take a look at carbon, again we see its valence is 4. It has 6 electrons total. Its atomic number is 6. 2 in the first shell, 1, 2, 3, 4 in the outer shell. All right, so it needs 4 bonds, or 4 more electrons brought in to complete its outer shell. That's why its valence is 4. Nitrogen has an atomic number of 7. 2 electrons in the first shell, 5 in the second, so its valence is 3. It needs to bring in 3 more electrons to get up to 8 in the second shell. Valence of 3 forms 3 bonds. Oxygen to its left, atomic number of 8. 8 electrons, 2 in the first shell, 6 in the second. It's missing 2 electrons in its outermost shell to be complete, so its valence is 2, always forms 2 bonds. Hydrogen, valence of 1, needs 1 more electron, and it's only an outermost shell. Therefore, its valence is 1, always forms 1 bond. So, knowing this, if I were to give you a molecular formula for a compound, you should be able to deduce or logically figure out the structure of that molecule. Let's look at some examples. Okay, so here we have a molecule called urea. Urea has a formula CONH2, 2. All right. So knowing that carbon forms 4 bonds, oxygen forms 2 bonds, nitrogen 3, and hydrogen 1, with that particular formula, this is the only structure you can come up with. Let's see why that is. This carbon, does it have 4 bonds to it? Ask yourself that. Yes. 2 here, 3, 4. Nitrogen, does each of them have 3 bonds? Yes. This one has 1, 2, 3. This one, 1, 2, 3. Hydrogen, every single hydrogen has a single bond. Oxygen, 2 bonds. Every atom in that structure of urea there has its valence completed, has its outermost shell completed by bonding in the way that they do. And so it's the distribution of electrons in the outermost shell that determines how many bonds the atoms will form, and that determines the structure and the formula of the molecules that they produce when they come together. All right. What about a simpler example, say CO2, carbon dioxide? Well, knowing the valences, there's only one structure you can draw, and this is it. That's it. That's the structure of CO2. every valence is completed. Each oxygen has two bonds, giving it 2 electrons needed to complete the outer shell. Carbon has 4 bonds, giving it the 4 electrons it needs to complete its outer shell. All right. Now, let's start talking about the diversity of carbon compounds. So carbon compounds are made typically of carbon chains of carbons linked together. Okay, that's not true of urea that you see here, but in most cases it is true, as we'll see especially in the next chapter. So carbon chains form the skeletons of most organic molecules, and these carbon chains, carbons linked end to end, can vary in length and shape. All right, so I have an animation here. Let me play this real quick.
The carbon backbones of organic molecules may vary in length and in location of single and double bonds. Molecules may be straight or branched and may even contain rings of atoms.
Okay, so we have lots of diversity of molecular structure possible because of the way carbon chains can form, and that's sort of what we're going to focus on here in much of the rest of this chapter.
The carbon
Let's take an example here. So this is showing you some of the diversity that we can get in carbon chains. Now, basically, there are 4 or 5 different ways that carbon chains can vary, okay? Sort of variables that go into making them different from one another. What I call facets of diversity, right? Or facets of variation. So first we have length, simply the length of the carbon chain. Look at ethane, C2H6. Has certain chemical and physical properties as a result of its formula being C2H6. Propane, C3H8, has different chemical and physical properties than ethane. Even though it only has 1 extra carbon and 2 extra hydrogens, it's very similar, straight carbon chain, all that. These two are very similar, but they have very distinctive properties simply because they have different numbers of the same types of atoms. All right, especially having different numbers of carbons. Second facet of diversity here is branching, whether we have a linear or straight carbon chain, as in butane, or a branched chain, as in 2-methylpropane. Now, look at butane. Butane has a molecular formula of C4H10. 2-methylpropane, also C4H10. They have the same chemical formula, but while butane has a linear or straight carbon chain, 2-methylpropane has a branched chain, and that alone gives them different shapes and structures and therefore different chemical and physical properties. They're different simply as a result of branching versus linear. Third facet of variation, double bond position. Here we have 1-butene. Notice again the ending ENE. That's telling you there's at least 1 double bond somewhere in that carbon chain. 2-butene has it here. So the 1 right there is telling you the double bond comes off carbon number 1. That's number 1, there's 2, 3, 4. 1-butene. 2-butene. Double bond comes off carbon number 2. So you see how the name gives you information about the structure of that molecule. 1-butene and 2-butene having the double bond in different position will have different chemical and physical properties as a result. Keep in mind this idea of structure and function, right? Okay, fourth, we have presence of ring structures, cyclohexane. So ANE ending, all the carbon bonds are single bonds. Cyclo, meaning they're formed in a ring structure, cyclohexane, okay? Now in this case, the hex tells you there are 6 carbons. You don't always have a prefix like that telling you the number of carbons, but you do here. This is the abbreviation for that. Every angle that you see, if there's no letter there, that indicates it's a carbon. If you see a letter there, like an O, that's telling you, okay, it's not a carbon, it's oxygen. But here you see the abbreviation, no letters, so it's this. Here we have benzene, ENE ending. You know there's at least 1 double bond. It actually has 3. Now notice it has fewer hydrogens. Why is that? Because their bonding capacity is taken up by double bonds in the carbons, there's less room for hydrogen. This carbon has 1, 2, 3, 4. This one has 1, 2, 3, 4, and so on. That's the abbreviation for it. So you see that just with these 4 facets of variability, length, branching, double bond position, presence of rings, we can have a lot of diversity of molecular form and function. Okay, Let's talk briefly about a special category of organic molecule, and that is the hydrocarbon. Hydrocarbons. Hydrocarbons are organic molecules consisting only of carbon and hydrogen, or at least they have portions of the molecule that consist only of hydrogen and carbon. Fats are a great example. Fats and oils are great examples of hydrocarbons. One of the things about hydrocarbons is that they are extremely good at storing potential energy. This is why fat stores so much more energy than, say, sugar does, because sugar is not a hydrocarbon, fat is. And so what you have in a hydrocarbon is a long chain of carbon atoms linked together, and they have nothing but hydrogen around them. And that hydrocarbon stores a large amount of energy. Okay, and we'll talk about hydrocarbons as fats in particular more in the next chapter. But if we take a look at an example here, here you see a fat molecule. That's a hydrocarbon. This portion technically is not hydrocarbon. What is? 1, 2, 3 carbon chains. These are called fatty acid chains. The black area in the middle is the carbon chain, and we have hydrogen atoms in gray attached to it. This is sort of like the fuel tank of that molecule. Those 3 hydrocarbon chains attached to that other portion of the molecule means this is a fat. It stores a lot of energy. On the left, you see a microscope view of what's called adipose cells. Adipose cells are specialized mammalian cells for storing fat, and you see all these fat droplets here inside that. For instance, there's the nucleus of the cell. There's an outer membrane here you can't see in the photo, but these are all fat droplets. They're composed of hydrocarbon. So, mammal fat cells store a large amount of energy. Okay, next thing we're going to talk about is sort of another facet of diversity that we can find in organic compounds, and that is the concept of isomers. Okay, isomers are compounds with the same molecular formula but different structures and properties. All right, different structures and properties. We have basically 3 different categories of isomers. We have what are called structural isomers, which we'll talk more about in a minute. We have cis-trans isomers, okay? And we have what are called enantiomers. Enantiomers. So, briefly, structural isomers are molecules that have the same chemical formula, but just a different arrangement of the atoms, okay? Cis-trans isomers have the same chemical formula and the same covalent bonds, but they differ in their spatial arrangements. If that's not clear, you'll see what I mean in a minute. And then enantiomers are isomers that are mirror images of one another. So all of these different categories of isomers involve molecules that have the same chemical formula, same number and type of atoms, but they're categorized based on how those atoms are arranged, okay, in the 2 different molecules. So let me play this little animation. This will help explain some of this.
3 types of isomers are structural isomers, which differ in the covalent partnerships between their atoms, geometric isomers, which vary in arrangement of atoms around a double bond, enantiomers, or stereoisomers, which are molecules that are mirror images of each other, like left and right hands.
Okay, so let's move on to the next slide, and I'll explain this in a little more
3
detail.
types.
All right, so in part A up here, we have an example of structural isomers, pentane and 2-methylbutane. So pentane is C5H12, that's its formula. But count the carbons and hydrogens. C5H12 is also the formula for 2-methylbutane. But you see this has a linear carbon chain or a straight chain. This one has a branched chain. And actually, the 2 there tells you that the branch comes off carbon number 2. So they have the same chemical formula, but a different arrangement of those atoms. These are the simplest types of isomers. They are structural isomers. Now, in the case of cis-trans isomers, again, same chemical formula, but notice here you have X. Okay, so that X, these X's represent an abbreviation of some larger, more complex grouping of atoms. Okay, that X could be 5, 10, 15, 20 different atoms, so we abbreviate it as X. There's a duplicate X group right here. In the case of a cis isomer, which this one is, those two X groups are on the same side of the carbon chain. They're on the top here. That's what cis means, same side. Notice over here, same structure, trans, right? C2H2X2, C2H2X2. But in the trans isomer, the X groups are on opposite sides of that carbon chain. That's what trans means. Think about transcontinental, across continent. Think trans here, across the carbon chain. They will have different chemical and physical properties simply as a result of those X groups being in different arrangements relative to the carbon chain, cis or trans. Now here we have enantiomers. These are mirror images of each other. So notice we have carbon, oxygen, hydrogen, a CH3 group, NH2. Carbon, right? I'm sorry, that's not oxygen. So we have carbon, CO2H, H, CH3, NH2. We have the same groups attached to the carbon over here in this case. One of them is called an L isomer, the other is called a D isomer. They are mirror images of each other. A lot of times students will look at these and they'll say, well, you know, you could just take this one and rotate it 180, like turning your hand around so you see the back of it in this case, and that would give you this one. It won't. Yes, if you rotated this molecule, right, if you rotated this molecule, the H would end up over here, the NH2 would go over there, and that would match up, but the CH3 group would rotate around to the back, whereas here it's on the front. You cannot rotate either one of those to make them match up perfectly with the other. In the same way, you cannot rotate one hand to make it line up perfectly with the other. If you rotate your hands or hold them out in front of you so you're looking at your two hands, the back of one facing you and the palm of the other facing you, yeah, your thumbs and pinkies are on the same side, but again, you're looking at the palm of one and the back of the other. You cannot rotate them. Try it. You cannot rotate them to make them match up perfectly because they're mirror images. If you were to put a mirror right here in the center so that the image of this molecule hits that mirror and bounces off to your eye, the image that you would see here in the reflection is not of this one, it's this. The reflection is the mirror image of that molecule. So they are mirror images of each other, and you might not think that this would make a big difference, but it can make a huge difference in terms of the characteristics of these molecules. Enantiomers are really important in the pharmaceutical industry. Two enantiomers of a drug may have entirely different effects, okay? Usually only one isomer, one enantiomer, is biologically active, but typically both are produced just as a result of the way that the chemistry works, and they have to isolate the effective one from the one that doesn't do anything through a complicated process. And you go to the pharmacy, buy the drug, you're getting the effective enantiomer that's been isolated or separated out from the non-effective one. And so these differing effects of enantiomers demonstrate that organisms are sensitive to even subtle variations in molecules. Let's look at an example here.
Isomers are compounds that have the same molecular formulas but different three-dimensional structures. The difference between isomers can literally mean the difference between life and death. L-DOPA, shown on the left, reduces the symptoms of Parkinson's disease. Its isomer R-DOPA on the right has no effect on the disease.
All right, and another example of that, here you have ibuprofen. This is a common painkiller. I think it's the primary painkiller in Advil. If you take Advil for a headache or whatever, reduces inflammation and pain. It comes in two forms, S-ibuprofen and R-ibuprofen. R-ibuprofen, the mirror image of S, doesn't do anything. It's not effective as a painkiller. S-ibuprofen is effective. This is the one you get in the bottle of Advil when you go and buy it at Target, for example. Albuterol, which is a medication that people take for asthma. It relaxes bronchial muscles, improving airflow. It's in the inhalers that you see asthmatics taking occasionally or inhaling from. R-albuterol is the effective version of the molecule. S-albuterol doesn't do anything. So what you're getting in the inhaler is R-albuterol. This really exemplifies this idea, this concept of structure and function. Things work the way that they do because of their structure, because of their shapes, okay? Even subtle differences, mirror image, left versus right, can have dramatic effects. Okay, so enantiomers are yet another faceted variation that can result in structure and function of organic molecules. Let's talk about another one. We have a few chemical groups that can be attached to a carbon chain that are key to understanding the functions of molecules, okay? And these chemical groups are called functional groups. Functional groups. If you take organic chemistry, you'll probably study about 25 or 30 different functional groups. In biology, however, we care about 7 of them. There are 7 of them that you find routinely in sugars, proteins, fats, and nucleic acids. And each one of these functional groups has its own distinctive characteristics. So if you're looking at a molecule, say a structural formula for a molecule, and you see a certain functional group, if you know what the functional group, or the characteristics rather, that functional group are, that can help you understand the functional characteristics of the molecule that they're attached to. The functional groups sort of impart a flavor or a characteristic to the molecules they're attached to, in other words. All right. Now, as an example of how functional groups can impart characteristics to molecules, think about estrogen and testosterone. So estrogen is also known as estradiol, okay? And let me get to that slide. So we see estrogen or estradiol here. We see testosterone here. These are two of the primary sex hormones in vertebrate animals, okay, like ourselves. Estrogen is primarily responsible for female secondary sexual characteristics. When I say secondary sexual characteristics, I don't mean the obvious things like genitalia differences. I mean overall body form differences, right? For example, if I were to show you just the silhouettes, the outlines of a male and female human, you would probably be able to tell which was male, which was female, based on females having wider hips, males having more narrow hips, males being taller with broader shoulders, females being a little bit shorter in stature with more narrow shoulders. You get the idea, right? Those are secondary sexual characteristics, and these are primarily sort of controlled by estrogen and testosterone. So you get very different effects of these two molecules. But overall, look at them. They have very similar characteristics, structures. 1, 2, 3, 4 carbon rings all fused together. You basically see that in testosterone as well. What's primarily different are the functional groups attached to them. Here we have a functional group called a hydroxyl group, which we'll see more in a minute, right, on estrogen. You don't see it on testosterone. That's just an oxygen. Here you have one called a methyl group on testosterone, not on estrogen. This is the same between the two. So it's some very subtle differences between these two molecules that give them impart to them very different characteristics, and therefore they have very different effects. Take a look. These are secondary sexual characteristics in male and female lions, right? Male, much larger in body size, has a mane. Female, smaller body size, no mane. You can tell which is the male, which is the female from 100 yards distance. But those differences that you see there are primarily a result of those subtle differences between estrogen and testosterone, which again are due to the functional groups, okay? So, functional groups are the components of organic molecules that are most commonly involved in chemical reactions. Think of a functional group as like the business end of a molecule. When two molecules bump into each other, if their functional groups are aligned when they bump into each other, you can get a chemical reaction between those functional groups. It's why they're called functional, okay? That's where the chemical reaction between two molecules will occur, at the functional groups. And it's the number and arrangement of functional groups that give each molecule its unique properties. So what functional groups are we talking about? Well, there are about 7 of them. And let me just show you this figure here. There's a lot of information on this figure from the textbook, but you don't need to know most of it. Okay, this is really meant to just show you some examples of these. All right, so first of all, we have what are called hydroxyl groups. Hydroxyl is simply an oxygen bonded to a hydrogen. right there. You find these in alcohols. You find them in sugars, for example. One thing about them is that they are polar, and that's because that oxygen pulls the electron away from the hydrogen, giving it positive charge here, negative charge there. So this is why alcohols, sugars, and things like that dissolve in water. That has a charge on it, as you see here in ethanol, and this is why ethanol will dissolve in water. It makes mixed drinks possible, all right? Carbonyl groups. This is a carbon double bonded to an oxygen, as you see right there, carbon, oxygen. These are found in a group of molecules called ketones and aldehydes. Not really important. Acetone, ladies, if you use nail polish remover, one of the primary solvents in it is acetone, gives its characteristic smell. It is a ketone. It has a carbonyl group right there. Okay, here's propanol, just another example. Carboxyl groups. These are really important. Carboxyl groups are found in a larger molecule called an amino acid. Amino acids are the building blocks of proteins, and every amino acid has one carboxyl group. It's COOH, a carbon double bonded to an oxygen, and that's single bonded to a hydroxyl. The whole thing is called a carboxyl group. Here we have acetic acid. This is the primary acid in vinegar, right? Carbon double bonded to oxygen, OH group, carboxyl group. Now, the reason why these are very often called acids, or on molecules called acids, is because you put these in water, the hydrogen dissociates from the oxygen, breaks away, as I talked about in chapter 3, and becomes a proton. And when protons build up in solution, they can drive pH down. So very often you see carboxyl groups on molecules called acids. Amino acids are the building blocks of proteins. We call them acids because they have carboxyl groups. We'll see more about that in the next chapter. Amino group, NH2, right there. Nitrogen, 2 hydrogens, right there. These make up molecules called amines, okay? Amino acids, I said, are building blocks of proteins. We call them acids because they all have a carboxyl group. We call them amino acids because each one of them has an amino group. Again, we'll see more about that in the next chapter, but here is a glycine, or an amino acid. It's called glycine. There's the amino group, there's the carboxyl group. Very often what happens is that hydrogen, again, breaks away or dissociates from the oxygen on the carboxyl, comes around and attaches to the NH2, the amino group, making NH3. And this reaction goes back and forth all the time, okay? But the point is, there you have the amino group. Okay, sulfhydryl. These are not as common. A sulfhydryl group is simply a sulfur bonded to a hydrogen, right there, sulfur hydrogen. You do find these in 1 or 2 amino acids, and as we'll see in the next chapter, when they come into contact with each other, 2 sulfhydryls, you can get a very strong covalent bond between them, and that's really the context in which we're going to talk about them in the next chapter. Phosphate group, PO4. These are critically important. It's a phosphorus with 4 oxygens around it. These are very good at storing energy, as we're going to see in a molecule called ATP a little bit later on, okay? But here you just see one example. This is glycerol phosphate group attached to a carbon chain. We're going to see plenty of examples of these kinds of molecules when we get into our unit on metabolism, where we're trying to understand how it is molecules store energy and then supply that energy to cells as they need it. And phosphate groups are very important for that. Okay, next thing we have are methyl groups. Methyl groups are CH3. CH3, we already saw one of those, or an example of those in testosterone and estrogen in the previous slide. Compounds that have them are referred to as methylated compounds. Here's one example, 5-methylcytosine. So methyl, right? There you see that methyl group, CH3. So at this point, it's not critically important that you understand all the characteristics of these different functional groups, but you do need to be able to recognize them. If I show you a molecule, for example, say like this one on a test, and I say, what's the functional group? You need to be able to tell me, oh, there's a sulfhydryl, okay, oh, there's a hydroxyl, there's an amino group right there. That's what you need to understand at this point. All right, so essentially that wraps up this chapter. We got through that all in one sitting. That's chapter 4. It's a fairly short chapter. In the next chapter, chapter 5, we're going to go into the 4 categories of biomolecules that are made of carbon atoms in a lot more detail.